So this data comes out of some studies I was doing in rodent metabolomics, looking at tissue metabolism, and what we're going to look at is how a mode of anesthesia and or euthanasia in sort of tissue collection strategy affects the metabolome, and sort of going back to the talks earlier this morning, talking about how sample collection strategy and everything is important. It's sort of a cautionary tale. Watch out for how sampling can affect your metabolome, but it's also an encouragement that in spite of sampling technique, we can really do a good job and get useful information out of all sorts of samples in metabolomics. So where are we in the workflow? In this case study, we're here in this sample collection, study design, sample preparation that's kind of been the theme of this first day of talks here. And so objectives, first, we should, in general terms, be aware of the challenges associated with sample collection for metabolomics. Kelly already put some of that in your mind. Hopefully you also come away thinking that, hey, I can do this. This is a challenge, something to be aware of, so that you get meaningful data and you don't waste your effort, but it's also something that we can handle and we can get meaningful data out of at the end of the day. And then hopefully you'll understand some of the specific effects of anesthesia and euthanasia on the rodent tissue metabolome. This is something that you might not have to think about so much if you're going to look at you look at the genetics, look at gene expression, or even protein levels in an animal, but is very relevant to the metabolome. And then hopefully be able to select tissue collection strategies for those of you that are going to look at tissue. The tissue metabolome in order to reduce metabolic perturbations. Okay, so overview, we'll talk about metabolomics of tissues, what makes it unique. We'll look at the purpose of this particular study. I'll talk methods, tissue collection methods, and sample and data analysis methods for this study. I will look at some results in terms of the effect of anesthesia on the global metabolome in an untargeted and in a more targeted way, and then we'll make some recommendations. How would you collect rodent tissues for metabolomics, and what about human samples for metabolomics? Okay, so sample preparation. So why are we spending so much time on this today? You'll be sick of it by the time you're done. Well, again, as Maureen was telling you, substantial time will be spent designing and performing your experiment, both on your end and on the core lab's end, analyzing the samples and interpreting the data, but the results could be anywhere from very meaningful to entirely meaningless if the samples are collected in a way that don't reflect the biological conditions you want to study. So we've found that metabolomics of tissue samples from living animals is particularly prone to changes induced by a non-ideal tissue collection strategy. No sample type is immune to this, so we're going to tell you about how to do that properly, or at least as close to properly as we can, and that's where we'll be. So, okay, metabolomics of tissues and organs. So why is metabolomics of tissues important? Biofluids, blood, urine, CSF, things like that are great because they represent an average metabolic state of the organism, and they're a great place to start when you're doing a study. They're readily accessible. It's easy to get blood, minimally invasive, easy to handle, but it's difficult to pinpoint the source of the metabolic perturbation that you're studying. So if you're looking at blood and you see a change in lactate levels, is that coming from the skeletal muscle? Is it coming from the liver? Is it coming from both of them? You might not know. If you see a unique metabolite, you may not know what the organ level origin of that is. You just know that something has changed. So that's good, but oftentimes you want to get a more specific answer about what's going on. So tissues offer a more focused view of the metabolome. They're the site of metabolic regulation. They're where metabolism is most actively occurring, although some metabolism does occur in blood. Then they provide direct insight into what might be dysregulated in the disease that you're studying. However, they require more laborious collection and sample handling. It's easy to collect blood. It's a good bit more tricky to collect a muscle biopsy or to collect tissue out of an animal. Okay, so the purpose of this particular study, many aspects of sample preparation have been thoroughly studied. When you look at the literature that's out there, People have done to no end studying the difference between serum and plasma. Maybe that's a question you have. Should I collect serum or plasma? What anticoagulant should I use if collecting plasma? My answer to that question, just briefly, is it doesn't make that much difference, serum versus plasma. We tend to favor plasma a little bit just because you can get it cold right away rather than having to leave it at room temperature and wait for it to clot. Clotting it at ice temperature is delayed and a little bit different of a process. But both of them work fine for metabolomics. The choice of anticoagulant, we've had decent success with both EDTA and heparin, and citrate not as good. I don't know. I forget if Kelly mentioned that earlier today. But generally, you can use, that's well studied in the literature. You can read about that. Choice of extraction solvent, as I mentioned, that's something that affects us, and that's been studied to no end. Analytical method, reproducibility, and sensitivity, linearity, all of those things are relatively well studied, as they should be. Sample collection strategy for cells has been reasonably well studied. There's lots of different techniques, and we have a well studied technique and something that's available on our website as far as a strategy that you can use to collect samples and to quench metabolism for cells. There's previously, up to the point that we did this study, there was less information about the effects of sample collection for rodent tissue metabolomics, including the important but often ignored steps of anesthetizing or performing euthanasia on animal prior to tissue collection. And most literature that's available focuses on assays that are less subject to short-term alterations. In other words, they look at the genes or gene expression or protein levels, things that may not have quite the same speed of response as the metabolome does. So the goal of this study was then to examine the effect of 6 commonly used methods of anesthesia and or euthanasia that you might use when you're dealing with your animals in the lab on metabolome and devise a recommended strategy. And so this reference is available. We published this in PLOS One a few years ago, a couple years ago now, and you can look at this, the results from this study in detail. And just to state ethics, anytime you're doing with animal work, as you guys well know, you need to follow appropriate ethical procedures and have your procedure. This is now the Institutional Animal Care and Use Committee. All of this was approved by that. So here's a workflow. We took C57 black 6 mice, probably the most studied animal model that's out there as far as rodents are concerned. And we collected different tissues. We collected skeletal muscle. We collected plasma. We collected liver. We collected heart tissue. And we collected white adipose. And we, after performing 6 different modes of anesthesia and euthanasia, all of which are commonly used in the literature in working with these animals. And then we perform metabolomics, and as Maureen had mentioned, and others who talked today, the first step with metabolomics of tissues that we generally will perform when you have enough of it to work with is to cryopulverize them. So we keep this stuff frozen. We grind it to a powder to aid in extraction. We'll weigh out a specific amount of it that we need. We'll add an extraction solvent. We'll sonicate or do some other tissue disruption strategy. If we're doing really high throughput, we can do a homogenization strategy that puts all of this into one step. We'll pellet the precipitated protein and other sample debris. We'll collect our supernatant containing the metabolome, run it on the LCMS, and then we did targeted and untargeted metabolomic profiling to look at differences. Okay, so what were the methods of anesthesia and euthanasia that we studied? So the common ones that are performed in the lab. These were 20-week-old mice, fasted 5 hours. For mice, a 5-hour fast is roughly equivalent to an overnight fast in humans. When you go for overnight in mice, it gets more towards a starvation condition. But you know, basically just trying to be consistent, sample the animals at the same time of day, after the same sort of handling procedure, the same period of acclimation, in order to minimize the variability. And we did these standard methods of collection. So a lot of people, when they're collecting tissues, will euthanize their animals by a standard method. Cervical dislocation is the quickest. They might use carbon dioxide or an isoflurane overdose, and just collect your tissues after that necessary step is done. And so this results in various different times from initiation of the activity of the euthanasia to the time when you can safely collect tissues. Cervical dislocation is fast. That needs to be a specifically approved protocol if you're going to use it as your only method of tissue collection. Carbon dioxide is generally considered an acceptable method of euthanasia. It takes a while. though for the animals to be anesthetized and ultimately euthanized and be ready for tissue collection. Isoflurane is about equivalent if you're using it as a mode of euthanasia. We also did tissue collection under anesthesia. So in other words, we established a surgical deep plane of anesthesia like you might do if you were going to do some sort of survival surgery. But then we collected tissues under these conditions. Of course, you're collecting major organs, so this was a terminal event. But blood circulation was ongoing to the point of collection of the tissue. And this is a standard protocol that is sometimes used, but it's actually a lot less common, a lot less commonly done, and nothing had been really studied about whether this makes a difference at all in terms of metabolomics. Some of these methods are fast. Some of them, when you do an IP injection of these metabolized anesthetics, it takes a while for them to reach the cervical plane where you can do these things. Once anesthesia was established or euthanasia was complete, we did immediate dissection of these of the tissues as rapid as our technique would allow, in a specific controlled order. That's another important thing to point out. Collected the tissues, liquid nitrogen froze them as quickly as we could, and stored them at -80 until the time of analysis. So just looking at the specific tissues, some method notes. So the order of collection was gastroc muscle. So this was a representative muscle. We could collect various different other muscles from the rat. This is done because it's peripheral, and we wanted it, and we wanted under the modes of anesthesia we were testing, we wanted blood circulation intact as long as possible. So you can do a collection here, which although there will be some blood loss, will not disturb. There will still be ongoing blood flow as the animal is anesthetized as you go on to the later tissues. If liver is what you really care about, of course, and you want muscle as something extra, you might choose to begin with liver, and I'll show you a little bit about how that might affect things later on. But this surgical dissection happened within 1 minute. We tried this both as collection by dissection or collection by freeze clamping in situ, and we got similar results. Then we collected arterial blood from the descending aorta from the abdominal cavity. Next, we collected liver and heart, and so these were done. This can be done very quickly, a few seconds here, and the heart dissection can be done within an additional minute. Blood flow is maintained to some degree, at least to this point, in the anesthetized animals. And then last of all, we collected white adipose tissue. Again, if white adipose tissue, this is, although generally you think of adipose as being somewhat more metabolically stable, that's not at all the case. When you look at some metabolites, maybe less so in triglycerides, but in free fatty acids and in the central carbon metabolism, white adipose tissue, brown adipose tissue are both very metabolically active, and you can have an effect on the metabolome just as well here as you can on other tissues. So this was our procedure. The tissue extraction procedure, just to go through it in a little bit of specificity here for this study. As I mentioned, we cryopulverized the tissue. We transferred a defined mass to the tubes. Something on the order of 30 milligrams is what we use when we have as much as we could want. We can definitely go to much smaller quantities when needed. Or somewhat smaller quantities. Like there gets to be a point where it's just challenging to get everything normalized, no matter what approach you apply when you go too small. Then we add extraction solvent. In this case, we used a mixture of methanol, water, and chloroform. It contained a C13 internal standard mix to enable absolute quantitation of some metabolites. That was our solvent ratio. We did probe sonication for further tissue disruption beyond the cryopulverization. Collected our supernatant from centrifugation. And then we did, in addition to knowing the wet mass that we started with, we dried the pellet and measured the mass for normalization. We can also do a protein assay on that. If in general, dry pellet, when you have enough of it, a dry tissue mass or a protein assay give somewhat comparable results. If you have smaller quantities, the protein assay can be a little bit more reliable than a weight. The analytical method that we used, you've heard a little bit about this already, but was a Hillock approach performed on an aminopropyl stationary phase. It's very good for polar metabolites, works for some non-polars, but we were interested in it tends to be that the polar metabolites, those things associated with central carbon metabolism, are the most affected, the most rapidly changing metabolites in tissues a lot of the time. So we wanted to do a method that was compatible with looking at that. We did the study on a time-of-flight mass spectrometer, so it's compatible with untargeted analysis, but also good for targeted quantitation. So this was one of those blended, targeted, untargeted profiling methods, and we did full scan analysis, which enabled that. So let's look at some untargeted metabolomics. So this is sort of a foretaste of what's coming tomorrow as far as a way of looking at your samples. And you'll see these sort of plots in metabolomics papers all the time, a principal component analysis plot. And so what it's doing, and I'm not going to explain it as well as George and others talking tomorrow can do, but what it's doing is taking all of the data that you've gone and generated, your hundreds of thousands of metabolites, and reducing it, right? So instead of trying to look at one metabolite, then another, then another, then another, then see what changed, we say, given the global profile of the data, how does group A differ from group B, or condition A group differ from condition B? And it sort of breaks the data down into these principal components, which are abstractions, which sort of define axes of change in your data. And basically, I'm botching the explanation of this to a substantial degree, but effectively, the end result is that if your groups are separated, then there are significant differences in the metabolome as a whole that define those differences and that are causing them to be separated along these principal component axes. And you can compare the extent of that separation based by looking at your different groups. So we have a lot of different groups. This is more than typically appear on your principal component plots in a lot of cases. But we have our 6 different modes of anesthesia or euthanasia. CD is cervical dislocation, CO2, isoflurane continuous, isoflurane overdose, ketamine, and pentobarbital. And so you can see in the skeletal muscle, we have sort of 2 major groups of samples. We have this CO2 and isoflurane overdose methods. These are both euthanasia methods. Then we have this cervical dislocation, which was a more rapid means of euthanasia that's sort of in the middle. And then down here we have our 3 modes of anesthesia, all 3 of them overlapping fairly substantially. You look at the other tissues and we see something relatively, we see a different degree of separation, but we see a sort of a similar profile. We see cervical dislocation. We see CO2 and isoflurane overdose, grouped together. We see cervical dislocation somewhere in the middle, and then the modes of anesthesia, sort of grouping closer to one another. Ketamine tend to be the one that was closer to the CO2. These guys separate out. The continuous isoflurane and the pentobarbital were the most similar in terms of mode of anesthesia, and something similar in the other tissues. White adipose, a little bit less distinction between the tissues. Liver was very distinct between these modes. Serum, again, the same sort of thing. Overlapping modes of anesthesia and overlap between the modes of euthanasia. So right now we've established that there's a big difference in the metabolome between techniques, even though we did the procedures as rapidly as we could. And maybe this is unsurprising, but maybe it's something that would not immediately be something you think about. We have a big difference in the metabolome between animals that are where tissues are collected with blood circulation intact and breathing versus those where we've euthanized them and collected the tissues immediately thereafter. Let's look at so this is just another flavor of analysis, partial least squared discriminant analysis, which is so you'll hear more about that tomorrow. A supervised method where the differentiation is done with the algorithm being aware of the different groups rather than completely unaware. But the effect is relatively similar. The separation may be a little bit more clean in this case from one condition to another. We can pull out things that may separate one mode of anesthesia from another, but the overall pattern is actually fairly similar to what we saw in the principal component analysis. So we've got something going on here for real. And looking at it again a little more closely, this I already defined. The largest distinction is between methods involving anesthesia and euthanasia. And so what metabolites are responsible for these differences? What's driving these differences? So we looked at the results from our untargeted metabolomics, and we basically ranked our metabolites based on the order of importance in driving those differences. That's something you can do either using partially squared discriminant analysis, or you can do using another method of statistically ranking your features. And these are the sort of features we pull out. And so the metabolites that were different from condition to condition were different between tissues, although we see some things that are somewhat the same We see inosine monophosphate appearing in a couple of different conditions. We see some TCA cycle metabolites appearing in a couple places. We see the serum has a little bit different of a profile. We see hypoxanthine, some other, more unique to particular species. In adipose, we see some things that correspond to fatty acids. Not surprising, those are enriched in those samples. So we see a variety of different metabolites. So we wanted to take this and look at it in a more pathway-specific manner and try to make a little bit more sense out of what could be going on here. So we turned to targeted metabolomics, and it turns out that the method that we used for untargeted metabolomics is equally useful for targeted metabolomics, and so we quantitated 112 known polar metabolites using the same LC-MS data. And here we have confident identities for everything. We used C13 internal standards where we could when they were available, and everything else we did relative quantitation. And these are just some of the representative species that we get in a related metabolome. And so then this is the data that was output that we published in our paper. And so this is a heat map listing all 100 or so. This actually has a few of them omitted, a few of the less interesting metabolites omitted. This is only things that have statistical differences between one group and another. And this is too much to see on a slide, but we have basically here our different tissues across the top and our methods of anesthesia across the side. And what we can see is a pattern that's similar here. We have, so everything in this case was normalized to cervical dislocation, not because that's our preferred technique, but because it's the one that involves the least experimental manipulation, the least outside drug or other chemical agent in anesthesia. And we thought that would serve as a useful reference metabolome to compare the other techniques to. It turns out that it was actually definitely not the preferred technique for tissue collection. The largest distinction that you see is, again, between the modes of anesthesia and the modes of euthanasia. And you can go in and look at individual things that were changed. Some things are increased, are up following euthanasia versus anesthesia. Some things go in the opposite direction. And so let's look at them. So here's just sort of a zoom approach of that. We see many things that are significant, again, highlighting that there are large differences in the glycolytic pathway, pentose phosphate also, the tricarboxylic acid cycle nucleotides. Things like amino acids were not as dramatically dysregulated, lipids also not as dramatically dysregulated. So the extent of the effects that we observed were dependent on the metabolite classes that we were looking at. Let's look at a few of them in a more familiar sort of bar graph format. So in skeletal muscle, looking at hexose monophosphates, the modes of euthanasia induced higher levels of hexose monophosphates compared to the modes of anesthesia. And so what we think is going on here is, not surprisingly, that the tissues become hypoxic relatively quickly following a cessation of blood circulation and respiration. And so increased glycolytic activity occurs in the tissues to compensate for that effect and for that loss of oxygen. And it's not that the tissues are dead. It's that the tissues are responding. They're continuing to make ATP and do what they need to do in the absence of sufficient circulating oxygen. And so it depends on what you're looking for. If you're looking to study glycolytic metabolites in their native state, we feel like you get a much closer picture here. Under anesthesia than you do post euthanasia. Lactate, again, not surprisingly, is up according to the different modes of anesthesia. And here we see the largest increase in lactate, not surprisingly, observed after cervical dislocation, and that's because this induced strong contraction in the animal skeletal muscle in the process of carrying it out. So not surprisingly, we see the greatest accumulation of lactate. So if you were looking to measure native concentrations of lactate in skeletal muscle, this would be a very bad way to collect your tissue. One of these modes of euthanasia would probably be a lot more reflective of what's going on. Similar things we saw, interestingly, in succinate. Succinate is kind of, it turns out to be among other things, a marker of hypoxia. And so we see a very similar effect in succinate as we saw in the glycolytic metabolites. Interestingly, this is not observed across all TCA cycle metabolites. They don't move in lockstep. You see accumulation of succinate, whereas you actually go the other way around. Citrate is lower, is beginning to be depleted in euthanasia as compared to during anesthesia. So they show different patterns of change, and the effect that you observe depends on where you are in the metabolic pathway. In the case of nucleotides, if you looked at ATP in skeletal muscle, you see no substantial effect. So that might be surprising. Well, why are my euthanized animals not showing a change in the ATP concentration? Well, it's because there are mechanisms in place to defend ATP levels in spite of various states of hypoxia of your cells. And so where you do see changes are in the breakdown products of ATP. So AMP, ADP are much more, this trended, this was significant, are much more sensitive markers of disruption in these nucleotide levels. So ATP is converted to ADP and AMP. The levels of AMP are actually very low in resting skeletal muscle, but as ATP begins to be depleted and not resynthesized as quickly, you see that. And so in our modes of anesthesia, we began to see increasing accumulation of AMP. And if you looked at creatine, so I don't have it shown here, but creatine and phosphocreatine, the things that maintain ATP for a brief period of time in the absence of aerobic metabolism, you could see the disruption there also under anesthesia. After euthanasia, creatine was up and phosphocreatine was down. And so that was probably what was maintaining the ATP levels. If you waited long enough, you'll for sure see depletion in ATP, but by that time, then you're moving towards tissue that's undergoing cell cessation of respiration and cessation of normal metabolism. So we can do things, fun things with this data and plot it on a network. And one thing that we observed was that we saw a lot of changes, not just in these AMP levels and nucleotide levels, but also in their downstream intermediates. In the tissue, we saw alterations in these nucleotide levels. In the blood, we see the downstream products of nucleotide breakdown. So hypoxanthine, inosine appeared in the blood at higher levels than in the euthanized animals compared to the anesthetized animals, corresponding to nucleotide breakdown. And so there's interconnections, and you can make sense of this. Some things. In this case, this is a Metscape diagram where bigger hexagons mean up and smaller hexagons mean down. And so we see increased accumulation of some and decreased levels of others, depending on whether you look cervical dislocation versus the other techniques. We're not limited to only saying, okay, wow, anesthesia is different from euthanasia. Good to know. There are specific effects of anesthetics. Isoflurane turned out to be a really good method in most cases, but it did induce increased levels of aspartate. This is possibly due to alterations in mitochondrial aspartate and aminotransferase in the liver as a response to anesthesia, and isoflurane is believed to be metabolized in the liver. We see differences in levels of malate, relatively consistent between isoflurane and pentobarbital, but ketamine looked a lot more like our modes of euthanasia for some reason here. So there are unique effects of particular anesthetics that you need to be aware of. The magnitude of those effects was much smaller than the difference between anesthesia and euthanasia. So recommendations for tissue metabolomics. Tissue collected under anesthesia with blood circulation intact show reduced evidence of hypoxia. Surprise, surprise. But just to know, even if you do it quickly, it still has an effect compared to tissue from euthanized animals. In our hands, the fastest to induce and easiest to administer was continuous administration of isoflurane delivered by an anesthetic machine, which allowed fast and reproducible collection. But you still need to be aware of particular effects that your anesthetic might have. Regardless of the mode of anesthesia you used, it's important to document whatever procedure you used in your publication. It can have an impact, just so that if someone goes back and tries to reproduce it, they know how you did this. And it's surprising how many papers that talk about tissue metabolomics don't even think to mention this. Of course, we need to follow ethical guidelines, and if at all possible, consult with the core lab first. What about human samples? So this was a study on rodents. So obviously, if you're doing human tissue sampling, you're going to be a lot more constrained. You can't access liver nearly so easily in living humans as you can in tissues. Your sampling method is also more constrained. You need to do anesthesia. You're not going to euthanize a person before collecting their tissues. Sorry to even joke about that. So you're really constrained on what you can do. The good news is that hypoxia is not going to be as much of a problem because you're dealing with anesthesia and local anesthesia at that. But certainly you do need to be aware of the potential effects on the metabolome as they are removed. Speed of tissue removal and sample handling is still critical. Your procedure may call for careful dissection and rinsing of your tissue before you freeze it, and that may be fine for most of your sampling procedures, but you should be aware that in so doing you may induce changes in the metabolome just as a sample goes without blood circulation for a period of time, and you should weigh those benefits from the potential alterations of the metabolome versus doing a more immediate freezing and accepting contamination with blood, et cetera. You also must consider the presence of anesthetic and its effects on the metabolome. The question was asked about lidocaine. Yes, we see lidocaine or whatever other local anesthetic is being used. It can cause interference with detection of metabolites immediately adjacent to lidocaine in the chromatogram. Generally, we don't see large effects of lidocaine on the metabolome itself, so it hasn't been that large of a problem, but it still should be something we're aware of. And again, consult with the core. So that's it. So happy to answer any specific questions about that or about tissue metabolomics in general. Thanks again.